corticomedullary gradient. We introduced that last time. We drew the corticomedullary gradient. We simply put some numbers on the board. We said that the corticomedullary gradient was such that the concentration of ions in the cortical region of the kidney was in the neighborhood of 300 milliosmoles, pretty much isosmotic with the blood volume, pretty much isosmotic with the plasma in the blood. And then we started talking about the idea that we could lay out a gradient such that it went from 300 milliosmoles in the cortex down to about 1,200 milliosmoles deep in the medulla. That was the corticomedullary gradient. We just put those numbers on the board. We didn't explain in any way how that corticomedullary gradient was generated. And that's what we want to try to do today. We want to try to lay out the structure of the juxtamedullary nephron as it extends from the proximal convoluted tubule to the descending limb of the loop of the nephron, to the ascending loop of the nephron, to the distal convoluted tubule, and then hooking up finally with the collecting duct. And we're going to see that by regulating the kinds of channels and pumps that we put in different regions of the epithelial cells forming the juxtamedullary nephron, that we can now generate this corticomedullary gradient. We'll talk about how that's going to happen. We're going to start with a little bit of review from what we talked about yesterday on the proximal convoluted tubule, because that proximal convoluted tubule is going to lead directly into the descending limb of the loop of the nephron. Now, where in the proximal convoluted tubule we had those sodium-driven cotransporters, and we had the paracellular movement of water and ions. We're now going to find that the epithelial cells forming the descending limb of the loop of the nephron are different. These epithelial cells forming the descending limb of the loop of the nephron are characterized by both what they have and what they don't have. And what I mean by that is that these epithelial cells contain transmembrane proteins called aquaporins. Remember that aquaporins are water channels. But what these epithelial cells don't have is they don't have ion channels. So instead of ion channels, we're going to put aquaporins in these cells. Now remember, we've got a tube. We have a tube that's going to go from 300 milliosmoles down through an ever-increasing ionic gradient. And as these epithelial cells face an ever-increasing ionic gradient, we're only going to have aquaporins in these cells. So effectively, we're going to drain that tube through the aquaporins, leaving behind the ions. So in this way, we're going to see that the concentration of ions inside the tube comes to equilibrium with the concentration of ions outside of the tube at every step along the descending limb. But this equilibration is accomplished simply by allowing water to leak out. Then we come to the hairpin turn. We go round the hairpin turn and start up the ascending limb of the loop of the nephron. The ascending loop of the nephron, the first region here, deep in the medulla, is going to be called the thin segment. It's named the thin segment because these cells are pretty thin. When you look at them in the microscope histologically, they're pretty thin cells. So we call this the thin segment. But if we ask the question, what kind of channels do we have in here? We find immediately after going around the hairpin turn, we change the complement of proteins in the membranes. The complement of proteins in the membranes and the descending limb was aquaporins, and now we have ion channels. So now where on the descending limb we were draining water, we're now effectively on the ascending limb, at least in the thin segment, we're going to be draining out ions and leaving the water behind because there are no aquaporins. So again, we're now coming to equilibrium. But now we're coming to equilibrium simply by the movement of ions. But as we move up, as the fluid moves up along the epithelial cells lining the ascending limb, eventually we get to the point where we can't simply use the gradient to move the ions, and we now have to actively move them. We have to start invoking ATP-dependent pumps and transporters. And we're going to see that one of the major players here in the ascending limb is going to be something known as the sodium-potassium 2-chloride transporter. So the salt inside the ascending limb is going to largely be removed across the apical membranes by this transporter. We're going to put the sodium, potassium, and chloride into the cytoplasm. And then as we fill up these cells, particularly with sodium, we're now going to allow the sodium-potassium pumps to pump that sodium out into the extracellular space. So we're going to use an active removal of the sodium into the so-called interstitial space, the extracellular space. We're going to allow potassium largely to just run out by its concentration gradient, and chloride will follow. But now what we have done through these transporters and these pumps, we've now reduced the ion concentration in the ascending limb from around 1,200 milliosmoles at the hairpin turn, so that by the time we reach the distal convoluted tubule, the concentrations are actually below the interstitial concentrations. Interstitial concentrations are always around, in the cortex, are around 300 milliosmoles. But then we'll find in the tube, because of the pumps and the transporters, we can get concentrations down around 50 millimolar sodium, 50 millimolar chloride, reaching the distal convoluted tubule. In the distal convoluted tubule, we're now going to think of this as the last place we can reclaim or reabsorb sodium and chloride. It's kind of a fib. If you take another course, you're going to find that this activity spills over into the collecting duct. But I want to make things pretty clear, bright lines here. And the bright line here is going to be distal convoluted tubule is the last place we can reclaim sodium, and that's going to be done by apical sodium channels and basolateral sodium-potassium ATPases. So we'll hold on to this idea of moving ions across the epithelial cells of the distal convoluted tubule by this trick. And if we don't reclaim the sodium and the chloride here in this convoluted tubule, it doesn't get done, because then it spills into the collecting duct and goes to the outside world. But the distal convoluted tubule is not only the last place where we can reabsorb sodium and chloride, it's also an important control point. So we're going to see that there are epithelial cells in the distal convoluted tubule. These specialized epithelial cells have a name. They're called macula densa cells. These macula densa cells are actually a kind of sensory cell. What they're doing is they're monitoring the concentration of sodium chloride in the distal convoluted tubule. The concentration of sodium chloride in the distal convoluted tubule serves the role as being a kind of proxy for glomerular filtration rate. Now, what that simply means is that if the fluid is moving through the loop of the nephron too fast, there's inadequate time to remove the sodium and chloride, so sodium chloride concentrations out in the distal convoluted tubule are too high. So if we have high sodium chloride concentrations, that tells us the glomerular filtration rate is too high. We better throttle it back. So we're going to see that these cells in the macula densa recognize too much sodium chloride and in response release adenosine. The adenosine now is a local hormone. The adenosine binds to smooth muscles on the afferent arterioles. And the binding of adenosine on the smooth muscles constricts the smooth muscle, acts almost like a valve to reduce the flow of blood from the afferent arteriole into the glomerular capillary. And that way we regulate glomerular filtration rate. This process is known as tubuloglomerular feedback. And it uses this combination of sodium chloride serving as a proxy for the rate at which fluid is moving from the blood vascular compartment into the renal compartment, and using that information then to regulate smooth muscle on the afferent arteriole. Then finally, we're going to find, if we get time to do this, out in the distal convoluted tubule, where we're thinking of this somewhat inaccurately, but I think appropriately, that this is the final point of regulation of the reuptake of sodium. If we don't do it here, it doesn't get done. This is now going to be under hormonal control. The hormonal control is aldosterone. We've talked about aldosterone. Aldosterone is one of these steroid hormones released from the adrenal cortex. The aldosterone now, as a steroid hormone, binds to cytoplasmic receptors. and in turn now becomes a transcription factor. The particular kind of proteins that this aldosterone hormone is going to turn on in the genome is it's going to upregulate the sodium channels on the apical membranes and the sodium-potassium pumps on the basolateral membranes. So we can more effectively remove the sodium from the fluid in the distal convoluted tubule. And then we'll talk a little bit about this. Now, it turns out that in most of our lives, with the kind of sodium we have coming into our diets, we don't use this very much. But in our ancestral past, when there wasn't enough sodium in our diets, we would use this all the time to scavenge as much sodium as we possibly could so that we didn't have to have, we wouldn't be eating quite so much sodium. But in the world of processed foods, we don't have a sodium shortage. We actually have problems in maintenance of healthcare because of too much sodium. So, any questions?
Yeah, one just real quick, so I go into this understanding. Are you telling me that the descending and ascending limbs of the loop of the nephron are made of different endothelium?
So, no, let's do it again. I'm telling you that the descending and ascending limbs of the loop of the nephron are both made of epithelia. But what we'll see is that each of these cells is distinct. And so each of the cells can regulate what kinds of proteins it puts into its plasma membranes. And the epithelial cells lining the tube of the descending limb only put in aquaporins. Once you go around the bend and start into the epithelial cells of the ascending limb, now you begin to put in a whole mixture of things, but you no longer put in aquaporins. Now you start putting in ion channels, and as you go farther and farther up the line of the ascending limb, now you begin to put in active transporters.
So the only reason that question was because there's a concentration gradient in the medulla itself. So
Right.
We go a high concentration back to low concentration
Right.
on the ascending. Is it not going to try to pull water back into the...
It will try, but there are no aquaporins. So one of the major distinctions is descending limbs, aquaporins. Ascending limbs, ion channels, no aquaporins. Okay, and I'll draw it out. And right, yes.
Is the entire nephron wrapped around by the afferent artery?
So is the entire nephron wrapped by the efferent arteriole? No. The only place where we find the afferent and efferent arterioles are in the cortex, feeding blood into and out of the glomerulus. We'll see later, though, and I think what you're jumping ahead to, and we'll see this on Monday, is that long loop of the nephron is basically surrounded by something known as the vasa recta. And the vasa recta is an extension of the efferent arteriole, but it is a true capillary. It doesn't have any smooth muscle on it. It's a very thin-walled capillary, and where things readily move across it. So we'll come to this on Monday, and we'll describe the idea that the concentration gradients that we'll set up today also come to equilibrium with the concentrations of ions, and we'll find also with urea inside the blood in that blood vessel. So you're right, there is a blood vessel. It's called the vasa recta, but it's truly a capillary. It is not either an arterial afferent or efferent arteriole. Okay. Anything else? Well, let's draw this as the cortico-medullary gradient. And the juxta medullary nephron. So we'll use our sort of simplistic view of the kidney, sort of cut through the middle. I'll remind you that this green structure out here is meant to be the renal calyx, and that we distinguished regions within the kidney itself. We use this purple dotted line, and we said that thin veneer on the outside of the kidney was the renal cortex. And then we said that everything beneath that, all this structure, was the renal medulla. We then superimposed on this structure, the kidney, that specialized juxtaglomerular nephron, and we said out here in the cortex we had Bowman's capsule. We said extending from Bowman's capsule was the proximal convoluted tubule. We said that proximal convoluted tubule then extended into this tube here, referred to as the descending limb of the loop of the nephron. Some of you may have seen this loop of the nephron referred to as the loop of Henle. It's a term that's been used for centuries or more than a century. It was named after a 19th century histologist, a guy named Henle. Well, we're trying to get away from these eponymous names and describe this more functionally as the loop of the nephron. It's the descending limb of the loop of the nephron. It loops around here, deep in the medulla, at the site known as the hairpin turn, implying that's a very sharp turn, and then the tube continues, comes back to the cortex. This region here is known as the ascending limb of the loop of the nephron. And then we're out here in this region, back in the cortex, in the region called the distal convoluted tubule. This is a proper tube, and it's a tube composed of epithelial cells, hundreds of thousands of epithelial cells, all lined up one next to another. And where we're headed from here is to begin to recognize the different components of transmembrane proteins that we can find in different regions of this tube. But it's all composed of adjacently located epithelial cells. But now what I want to do is talk about the concentrations for the corticomedullary gradient. Always out here in the medulla, we have about 300 millimosmoles. Now, more or less 130 to 140 millimoles of this is sodium. Something like 130 to 140 is chloride. There's a little bit of potassium, a little bit of calcium, a little bit of urea. But most of it, I mean, if you add these together, that's 260, 280 out of the 300. Pretty much, this is isosmotic with the plasma in blood. It's pretty much the same concentration. But if we simply measure the ionic concentrations going from the cortex deep into the medulla, and I'll show you this as a discontinuous gradient, down here deep in the medulla, we can reach concentrations under extreme circumstances of about 1,200 milliosmoles. This is what we do as humans. If you look at other animals, you look at something like a pack rat in the outback of Australia, those animals have exquisitely long nephrons, loops of the nephron, and they can actually concentrate their urine to much, much higher concentrations, 1,200 milliosmoles. We're kind of stuck with this. This is one of the reasons we can't drink seawater. Seawater is too osmotically concentrated, and we can't extract the water from the high salt in seawater. Regardless, though, we have a considerable gradient. There is roughly 4 times as much ionic concentration down here as there is in the medulla. And if we look at this, and I'm going to lay this out as though it were a layer cake with different concentrations at each layer. Now, that's not quite right. because this implies it's a discontinuous gradient. But it's in fact a continuous gradient. So that if we look deep in the medulla, we find 1200, but I'm going to put a number on each of these bands. So we get from 1200 to maybe 1000 to maybe 900 to 700 to 600 to 500 to 450 to 300. Just to imply here that there's a gradient that extends from high concentration of salt down here in the medulla to low concentration of salt out here in the cortex. And now, embedded within this corticomedullary gradient, embedded with this structure is this tube of epithelial cells. This tube of epithelial cells extending from the proximal convoluted tubule to the proximal convoluted tubule to the descending limb to the ascending limb to the distal convoluted tubule. And then I'll draw it in here, but I want to put it in a different color. So coming across the medulla will be the collecting duct. But for our purposes today, we're going to limit our discussion to the epithelial cells and their components in the proximal convoluted tubule, the descending ascending limb, and out to the distal convoluted tubule. So let's try to put this in context. Last time, we talked about the epithelial cells of the proximal convoluted tubule. And we drew these in these two parallel box systems. And we said that this is the proximal convoluted tubule. And we emphasized the idea that there was water movement and ion movement between the cells back into the peritubular capillary, a proper capillary composed only of a single layer of endothelial cells. So this is the peritubular capillary. But we also said that another characteristic of these epithelial cells is that they contain on their apical surface these so-called sodium-driven co-transporters. And we said that we could bind an ion of sodium. And remember, we have plenty of sodium out here. Concentrations out here are upwards of 130 and 40 millimolar. So there's plenty of sodium. We move sodium in. And along with that sodium, we move glucose. Put it here. But then what we did is we then put in the basolateral side of this membrane, we said that there is a specialized transporter called GLUT2. This is a facilitated transporter. It allowed simply for the glucose to flow out and back into the blood. The only place where we have these sodium-driven co-transporters for glucose is in the proximal convoluted tubule. The only place we have them is here. So that if you don't use these sodium-driven co-transporters to remove glucose here, it doesn't get done. It moves through the descending limb and the ascending limb out into the collecting duct to the outside world. This becomes important, particularly before they could control levels of glucose. This becomes important in cases where blood glucose levels get very high. In cases where people have diabetes and they're not generating adequate amounts of insulin, so that and the insulin is important in extracting the glucose from the blood and putting it into muscle. In these individuals who have diabetes, glucose levels get really, really high. And when the glucose levels get really high, they're filtered from the glomerulus into the proximal convoluted tubule. And if we have more glucose coming into the proximal convoluted tubule, then we have transporters to accommodate it. That glucose is going to spill over into the descending limb and go to the outside world. It's why the urine of diabetics, before we could control the levels of glucose in the blood, the urine of diabetics was commonly sweet because of this sugar, the glucose, in the urine. And that happened because of the spillover. Then in the early 1920s, there were a group of people who recognized you could harvest insulin from slaughtered animals, inject people with that insulin, and largely give these human beings with, give people with diabetes a whole new lease on life. But that's the story of the proximal convoluted tubule. What I want to emphasize now is how this proximal convoluted tubule extends into this tube lined with epithelial cells. This tube lined with epithelial cells that I'm trying to draw here represents this structure here. This tube is meant to be the descending limb of the loop of the nephron. So this tube, this descending limb of the tube, is composed simply of epithelial cells. And the fluid, which came from the blood vascular compartment out of the glomerulus and was brought into the proximal convoluted tubule that we talked about last time, that fluid is now moving through the proximal convoluted tubule and entering the descending limb of the loop of the nephron. So the fluid now is moving in this direction. Now, this isn't a scale, so I'm going to try to have you imagine that these individual epithelial cells, stacked one next to another, are continuous all along this tube. And simply to put some numbers on this, because out here we're in the cortex, the concentration out here is going to be about 300 milliosmoles. But as we follow this tube down into the medulla, the so-called interstitial, let me put that in green, the so-called interstitial concentration of ions, the extracellular concentration of ions, the interstitial concentration of ions go from 300 milliosmoles here, under some extreme circumstances, can reach 1,200 milliosmoles. Now, it's a slow increase. We slowly increase the extracellular concentration as we move from the cortex down to the medulla, but recognize that as we take these tiny steps from one epithelial cell to the next, that each of these epithelial cells is facing an ever higher extracellular or interstitial concentration of ions. Now, one of the hallmarks of these epithelial cells is that they lack ion channels. But instead of ion channels, these epithelial cells now contain aquaporins. And the aquaporins are basically water channels. Now recognize what's happening. As we step from one epithelial cell to the next along the descending limb, the extracellular concentrations out here in the interstitial space are getting ever higher and higher and higher. The ions are trapped in here. They can't leave because there are no ion channels. But what can leave? Water. So what's the water going to do? It's going to run down its concentration gradient. And as it leaves ions behind. So simply by draining the tube through the aquaporins across the epithelium, draining this inside of the tube of water, the concentration of ions gets higher and higher, and the concentration of ions here come to equilibrium. Equilibrium with the interstitial space. So effectively what we're doing is we're draining this tube of water as we move down across the corticomedullary gradient, but the purpose of that is now to set up this gradient so that on the outside and have it come to equilibrium with what's on the inside of the tube. We're going to see a little later that we're going to exploit this corticomedullary gradient, particularly out here in the collecting duct, because you notice the collecting duct crosses that corticomedullary gradient, and we'll come to that probably on Monday. But what I want to emphasize now is the water concentration inside the tube is going down, and conversely, the ion concentration is going up. Then we come round the bend. So let's imagine over here that we're talking about the ascending limb. And we'll draw again this series of boxes. Trying to imply that we have a tube constructed of epithelial cells. The same orientation applies, and what I mean by that is this is medulla. So the concentration down here is 1200 milliosmoles. Where the fluid movement here was from the proximal convoluted tubule down into the descending limb, now the fluid movement is in this direction in the ascending limb. So down below so if we were to do this and extend it to here, the loop, the hairpin turn would be someplace down here on the floor. So we have this descending limb, the hairpin turn, and now we're coming up the ascending limb. Where these cells only have water channels, we now change the story over here in the ascending limb. Over here in the ascending limb, these epithelial cells contain ion channels. But remember, the extracellular concentration out here is like 1200 milliosmoles. So now what we can do is we can simply allow for ions to move down their concentration gradient through these cells into the extracellular environment. What these cells lack is aquaporins. So where these descending limb cells had aquaporins and could serve as a drain, there's no way to drain the water out here. So now we have high concentrations of salt inside the tube because we've come to equilibrium at all points. The fluid comes around the turn, now starts to go up the ascending limb. And at each point on this limb, at least particularly in this initial region, called the thin segment. In the thin segment, we're simply going to use ion channels, allow the ions to leave, and come to equilibrium with the extracellular environment. The story over and over and over again here is coming to equilibrium. We come to equilibrium by letting water leave on the descending limb. We again come to equilibrium on the ascending limb, at least initially, by allowing the ions to leave. But as the fluid moves up the tube, we'll find that this fluid inside the tube can actually get to lower osmotic concentrations than the outside environment. So that in the upper regions of the ascending limb, up here, we can have concentrations of about 100 milliosmoles, which come from about 50 millimolar sodium and about 50 millimolar chloride. Well, how do you manage to get ion concentrations inside the tube lower than on the outside? Well, you've got to put in some energy into the system. You have to pump these ions. So one of the major players here on these epithelial cells in the so-called thick segment will be these transmembrane proteins on the epithelial cells called sodium potassium, 2 chloride transporters. This is one of the major players we see in these epithelial cells. And as the fluid is moving by, these transporters are snagging sodium ions and potassium ions and chloride ions and moving them from the tube into the cell. And then on the basolateral side, they're now going to be sodium, potassium ATPases. I think you can see as we start moving sodium into these cells, this bucket will fill, and we need to pump them out. That's also true up here in the proximal convoluted tubule. Remember, these sodium-driven cotransporters are allowing sodium to come into the cell. The concentration of sodium inside the cell begins to eke its way up. And so now on the basolateral side, we have sodium, potassium, ATPases, which now pump the sodium back out again. But that's also true here in the thick segment. The major idea I want you to hold on to, though, is that by using these transporters and these pumps, that we can reduce the concentration of ions inside the tube well below the interstitial or extracellular, also known as the extracellular concentration of ions, down to around 100 milliosmoles. Yes?
What happens with the chlorine?
Chloride pretty much follows. Now that's a bit of a fib. There are a whole bunch of chloride transporters in here. There are bicarbonate chloride exchangers. I think there are even some hydrogen ion chloride exchangers. And there are a bunch of exchangers these epithelial cells we're not even talking about. But if you think about the chloride just following the sodium, that works pretty well. There will be some chloride channels that will allow the chloride just to follow the sodium into the cells and maintain a certain level of charge equilibrium. We're going to leave the idea that we have charge equilibrium. If you take another course in this, you're going to find that you'll actually have extra chloride inside the tube at some point. And that will also produce a transepithelial charge difference. So these stories get ever more intricate. And what I'm trying to do here is just lay out the baseline. And if you take another course, you'll get farther and farther into the weeds, and the weeds get pretty thick here in the kidney. Anything else? Yes?
So in the descending limb, you say that the ions come to equilibrium with the excess at the space outside. How do they do that without ion channels?
So the way to think about this is in the descending limb, there's no way to move ions. There's no pathway. We have these impervious pathways. We have palisades built. But what we do have is we have drains through the aquaporins. So we drain the water out but leave the ions behind. As we drain the water out and leave the ions behind, then it comes to equilibrium. The ionic concentration inside the tube comes to equilibrium with the interstitial space simply by allowing the water to leave down its concentration gradient. Yes?
Does that mean the concentration inside gets up to 1200 milliosmoles?
So the question is, does the concentration inside the tube down deep in the medulla come to equilibrium? Yes, it does. Turns out that a lot of that won't be just sodium chloride. A lot of that will be urea. And we'll see a little later that down here deep in the medulla, where we have 1200 milliosmoles, about 600 milliosmoles of that is just urea. But the best we can do by sodium and chloride transport is get to 300 millimolar chloride and 300 millimolar sodium. That's the best we can do as humans. And the remainder is made up of urea. But we're going to see here that urea plays a really important role in the final throes of concentrating urine in the collecting duct. But let's hold off on that for a little bit. Anything else? Okay, so we've now brought the fluid down the descending limb, up the ascending limb, and now we've arrived back here at the distal convoluted tubule. Now there's another point here of control, and I want to go to the upper panel and talk about a new form of control up here. And I'll label this panel as tubulo-glomerular feedback. In the distal convoluted tubule. Now, if you look in the upper panel, you can imagine that fluid, particularly over here on the right, goes up, and then at the top there will be the distal convoluted tubule. That's shown by that region in the cortex that links into the orange segment. What I want to do, though, is draw it a little bit differently this time around. And I'm going to show this region of the distal convoluted tubule as a kind of circle. And then I'm going to try to do these parallel lines, and then within this draw the so-called epithelial cells. Of the distal convoluted tubule. If we look at these epithelial cells in the distal convoluted tubule, they are organized, they're oriented in such a way that they come right smack dab up against both the afferent and the efferent arterioles. So to remind you of this structure from yesterday, we're now up in the cortex. And this is meant to be Bowman's capsule. This is meant to be the glomerular capillary. So the blood flow through here is through the afferent arteriole into the glomerular capillary and back out through the efferent. The cells in here in the distal convoluted tubule have a name. They're called the macula densa cells. The macula densa cells monitor sodium chloride concentration in the distal convoluted tubule. Let's go back to the upper panel. The fluid flow is in this direction. We can imagine out here, I won't draw the whole thing in, but this could be the distal convoluted tubule. The fluid is flowing into this region. This is the site of the macula densa cells. Now, what's happening, if we come back to this, is that we're monitoring this sodium chloride concentration out here, where the distal convoluted tubule enters the cortical region of the kidney. If the sodium chloride concentrations are high here, that tells us that the fluid in the tube has not spent long enough in the tube, and there hasn't been an adequate opportunity in time to remove those ions. So a high, you can spell it out here and say that a high sodium chloride concentration indicates a high glomerular filtration rate. Another way of saying this is that we're using the sodium chloride concentration reaching the macula densa cells in the distal convoluted tubule as a proxy for the rate at which the fluid is moving through the tube. Another way of saying that is if we go back up to the upper panel. If fluid is moving rapidly from the glomerular capillary into Bowman's capsule, it's then going to rapidly flow through the tube, and the amount of time the fluid has in the tube before reaching the distal convoluted tubule is inadequate to withdraw all of the sodium and chloride. So sodium chloride concentrations are too high. If, on the other hand, the rate at which fluid is moving from the glomerular capillary into Bowman's capsule If that rate's too slow, then there's a long time for these epithelial cells to work on that fluid and remove most of the sodium. So by the time we get up here again, if the glomerular filtration rate is too slow, sodium chloride concentrations are too low, and so we better speed things up. So let's look at how this might work. So this is meant to be the lumen of the tube of the distal convoluted tubule. And we're looking at sodium chloride concentrations. If sodium chloride concentrations are high, that will tell these cells, these maculodensa cells, to release a molecule called adenosine. Adenosine is like a local hormone. It's like a paracrine effect. The adenosine now will bind to receptors on what I'm going to show here is this snaky structure being the smooth muscle, the arteriolar. This is intended to be the arteriolar smooth muscle. On this arteriolar smooth muscle will be G protein coupled receptors. This is intended to be a G protein coupled receptor that binds adenine or adenosine. So it binds adenosine, and when it binds adenosine, that signals the smooth muscle through the G protein coupled receptor to constrict. So let's look at what we've done. Sodium concentrations are too high. That means fluid is moving too rapidly through the nephron. We haven't had adequate time to remove the sodium and chloride. The high sodium chloride concentration causes the release of adenosine. The adenosine binds to the smooth muscle, and now the smooth muscle contracts. What's that going to do to the rate of flow of fluid through the afferent arteriole into the glomerular capillary? It's going to reduce it. What's that going to do to glomerular filtration rate if we have less fluid in here? Especially, and this does happen, if this adenosine on this smooth muscle or on the efferent arteriole relaxes that. What's going to happen to the pressures in here? They're going to go down. If we reduce the hydrostatic pressure along the wall of the glomerulus, what's that going to do to the rate of movement from the blood vascular compartment into the renal compartment? It's going to go down. So that reduces glomerular filtration rate. So now we have a longer time of the fluid in the tube before it gets back to the distal convoluted tubule and the sodium chloride levels start to drop. So now where they had gone from a normal of 50-50 up to 75-75, now maybe they go to 35. That reduces the release of adenosine. As we release that relaxes the lack of a binding to this GPCR, causes the smooth muscle to relax, and renew the fluid flow through the glomerular capillary and renew GFR. So this is constant yin and yang, this constant feedback based on the release of adenosine onto the smooth muscle, regulating the blood movement through the glomerular capillary. That's tubuloglomerular feedback. The last couple minutes. So any questions there? If not, let me show you a couple slides. Finish this with a couple of slides. So this first slide is one that I'm not that fond of, but I'll show it anyway. It shows, what it's trying to show is the position of the cortex relative to the medulla, and it shows different kinds of nephrons. We've talked about this great long loop of this nephron. That's the juxtamedullary nephron. That's unusual. Only about 15% of the nephrons are of that type. The rest have these sort of short, truncated nephrons. But they do pretty much the same thing. But it's this long guy that's important for establishing the corticomedullary gradient. This is a picture I actually like. And what it's showing is the various components of the nephron. Up here, it's showing Bowman's capsule encapsulating the glomerular capillary. Fluid moves from the blood vascular compartment into the renal compartment at this site. We talked about this yesterday. That fluid then flows through the proximal convoluted tubule. That's where we reclaim much of the water and salts, all of the glucose and amino acids. And then the fluid flows into the descending limb of the loop of the nephron, moves around the hairpin turn, comes up the ascending limb, and now right here in the distal convoluted tubule, this region of the distal convoluted tubule is tucked in between the afferent and efferent arteriole. In the next image, let me show you this because it's a much prettier picture than what I had on the board, but it's the same idea, where this is the distal convoluted tubule. This is the macula densa cell. It's tucked in between the afferent arteriole and the efferent arteriole. When this sees high sodium chloride concentrations, it releases adenosine, causing the smooth muscle on the afferent to contract, and to a lesser degree, the smooth muscle in the efferent to relax and reduces pressure. When the salt concentrations come back to normal, we then bring the adenosine release back to normal, and we now throttle back on the contraction of that smooth muscle around the afferent arteriole. And that's the summation of what happens out there. And next time, we'll talk about aldosterone and the final